In this short video, I am going to discuss the cell proliferation protocol, which is part of the cell biology protocol collection. The protocol is designed to measure the population of proliferating cells, which is based on a two-die experiment. First, we are going to select the protocol by going to the count size grid. Next, we'll push the protocols button and select the cell biology collection. Next, we'll scroll down to the cell proliferation protocol and press the load button. Each protocol includes a demo image or set of images. The protocol is set up by using the associated demo image. If you're using your own image set, you may modify the protocol as needed and save it for later use. To start, I'm going to select the drop-down by the reset button, select the load demo with the open demo image checked. Next, I'll press the sync button, which will read in the individual channels from the image. This is a multi-channel composite image. We'll use DAPI to define the cells, which are labeled with nuclei, and the GFP channel for measuring or identifying the proliferating cells. Next, we'll set up and test the protocol with the interactive button checked. Press the next step button. In this case, we're using intensity-based segmentation to identify the nuclei. These are well identified. We may also use smart segmentation if we need to use machine learning. In addition, there is a series of segmentation options. We have a minimum size, which can be used to filter out any debris or broken up cells, anything like that. And this can be used to not only separate the minimum size, but also filter out a larger size. Next, we can apply clean borders. The protocol has those applied. However, we can turn that off if you want to measure the cells touching the edge of the image. In this case, we're only looking if there's a positive, negative, where there are proliferating cells. So I'll turn that to none. Fill holes is turned off if there are any gaps in the nuclei that would fill those. And boundary shape, we're turning on object splitting. We also have watershed and none. Anytime I make a change in the segmentation options, simply press the apply button to see the results, and now I've picked up the nuclei along the edge of the image. We also include an interactive step if your particular sample requires you to stop at any particular step during the protocol, and you're running a batch processing mode, analyzing a multi-well plate or a folder of images. This will stop at that step. Next, we'll press the next step button. And here we're looking for GFP in the boundary of the nuclei. Again, we can use intensity thresholding or we can use smart segmentation machine learning to identify our cells of interest. In this case, do that. Here we can clearly see there are positive and negative for cell proliferation. So when I'm satisfied with my segmentation, I can press the OK button. Pressing the next step button, we can identify which cells are proliferating. In this case, we'll report a heat map, which allows you to display on a per well basis for multi-well plate data. We also show the image with the snapshot image with information regarding the analysis. In addition, we have a data table including a snapshot image, the number of cells, the number of proliferating cells, and the percent of cell proliferation. Once you have set up the protocol to your satisfaction, and if you've made changes, you can simply press the Save As button, and that will save a new protocol under the cell proliferation group with your own naming convention. When we're finished, press the Finish button, and now we can choose to either analyze other additional images if they are open, or we can do a batch process by simply taking off the interactive mode. And if it's a multi-well plate, you can select the wells and the rows that you would like to measure and run selected, or we can also point to a folder of images and do a batch process. And once you're completed doing your analysis, the data they completed the analysis, the data may simply be exported to Excel.